At the end of this video, you will understand how one of the most widely used recombinases, Cre, works. This will allow you to understand how recombination sites, or LoxP sites, are used to produce inversion, insertion, and excision reactions. And finally, you will be able to understand how we use mutations within these recombination sites to favor one reaction over another. So let's grab a pen and let's go to the whiteboard. Recombination can happen through homologous recombination anywhere in the genome, or through site-specific recombination. The latter is used by Cre. Cre recognizes a 34 base pair recombination site, which consists of two binding sites that are bound by Cre, and also a central spacer. Because the binding sites are inverted repeats, they do not have a directionality. And so the only directionality comes from the central spacer, and we can draw it by an arrow. In which direction the arrow is facing is actually just a convention. There is no biological meaning behind it, of course, because the sequence does not really have an arrow attached to it. But there is a convention in which direction we have to draw it. But a mechanistic importance only comes once we look at a second recombination site, and in which direction these two arrows will look relative to one another. And so let's draw a second recombination site. And we can again indicate the directionality. And for now, let's assume that these two recombination sites are anywhere in the genome. We don't care really where they are, so I will just flank it by some random DNA sequence. And we will now have a look first on how these two recombination sites would recombine, and then how these strands would have to be connected after recombination. So everything I'm going to tell you now is true for both Cre and Flip, because they're both tyrosine recombinases and they have roughly the same mechanism. But for simplicity, let's just talk about Cre and LoxP sites. So let's start at the beginning. We have here a LoxP site, and also on the bottom there's another LoxP site. And as we learned, each is bound by two Cre monomers, so one here at the binding site 1 and the other here at binding site 2, and the same for the other. So they need to find each other and form this tetramer. And it's also called a synaptic complex or synapses. And there's one interesting feature about it, and that is that there are multiple possibilities how they in theory could form. So the strands could look in either anti-parallel alignments or in parallel alignments, but what people found is that the actual structure is an anti-parallel alignment. So both Flip and Cre will have this arrangement where one LoxP site or FRT site looks in one direction and the other one in the other direction in this complex. Now let's go through it. So briefly, or what's important to know is that there are always just two subunits active. So you see this Y here. This is actually the tyrosine, so the active nucleophile in Cre and Flip. That's why they're called tyrosine recombinases. And as you can see, they're circled, the active ones. They're only two active at a time, and they will switch later on in the second part of this reaction. And that also means that there's never a double strand break. So there's only one strand cleaved at a time. And the reason for only two being active is simply in the structure. So it's not actually a tetramer, it's more of a pseudotetramer. So there is a break in symmetry, and it is more of a dimer of dimers. So you have one dimer here and one dimer here. And you see a little bit of less space between this dimer and more space between here. The details about why there is just two being active at a time is a bit more complex. But in short, the reason is that the tyrosine here is actually not coming from the same monomer here in case of Cre, and it's very similar for Flip. But actually, the tyrosine of this monomer is donated to this one, and the tyrosine of this monomer is donated to this one, and this one to this, and so on. So it's like a circle. And because this interface is very small, the tyrosine here can be successfully donated to this one, and it can be active. But because the space is too large here, this one will not receive an active tyrosine. This one again will receive one, and here again, this one won't. And so the space between those subunits will then determine which ones are active and which ones are not. And so we have here this, and it's actually also annotated here as type 1 interface and type 2 interface. So we'll first have a nucleophilic attack of the tyrosine, and it will cleave the strands here and here. Then, in a second step, the hydroxyl group of the nucleotides here, or first actually the strands will change, so they will go over here, and then the hydroxyl group will finally attack the phosphate here, and now the tyrosine is the leaving group, and you have this this arrangement. This is here a holiday junction, and we need to think about this holiday junction for one more second in the following way. When this holiday junction is formed, what you can see is that this darker strand will have to base pair with this lighter strand, and this is actually the region of the spacer, and the same goes for here. They need to base pair, and we can appreciate that a bit more over here. The top reaction here, we can ignore the lower one, is essentially the same, and what we see is there is the tyrosine, it will attack, and then the strands are going to exchange here, and then the hydroxyl group, which is not annotated here, will again attack the phosphate and displace the tyrosine. And then we have this holiday junction, and then it will go on. However, we can appreciate that the red strand, so the red spacer here, this is just a spacer sequence, will have to base pair with the blue one, and vice versa. So an important requirement for spacers of two recombination sites is that they need to be the same sequence. Only if the spacer has the same sequence, then this holiday junction can be resolved because there is successful base pairing. Alright, so we can only continue this reaction if the spacers are base pairing. There's one more thing we need to discuss, and actually that's where the cutting points are. So we can also appreciate that here, this is the spacer, here they are dots, because the sequence is indeed flexible as long as it's matching. And Cre and Flip cut a bit different, but they cut within the spacer. So Cre would be one nucleotide in here and also here, and Flip would cut outside of the spacer. The holiday junction can now be resolved, and this happens by an isomerization step. The interfaces change, and now the other two subunits are active, so the tyrosine is circled again. They cut the strands, they also have then here the strand exchange again, and then the hydroxyl group will displace the tyrosine, and we end up with the final recombinant products. So the mechanism might be a bit complicated, but it's really just two important steps that you need to remember. First, the two recombination sites pair in an anti-parallel alignment, as indicated by the arrows. And second, the strands are rearranged in this manner. And this is all you need to predict any recombination event. Let's have a look. Let's start with the first example. Let's assume we have the following arrangement up there. Always start by drawing the synapses first. So just as we learned, we have one recombination site here and one down here. And we're going to indicate the directionality with arrows. And then we just connect the strands as they are up here. So we start from the left, encounter the recombination site, and then according to the arrow we need to draw them like this, and that's the result. And now the third step is simply to reconnect the strands how they would end up after recombination. So we can start from here over here, and the same thing happens over here. And I think you can already see what the end product will be. it is an excision. Let's start with the second example. So in this case, we have two different DNA molecules, for example, a genome and a plasmid. I have written here the word to indicate the directionality or the orientation of the recombination product. You will see why this is important. And again, we draw the synapses. And we connect the strands as they would be up here. It's very easy now. And of course, now you have to recognize that the orientation of the plasmid is in the other way. So we would have something like this. And now let's reconnect the strands. And so the end product you see is we start over here, we move along here, and we encounter the plasmid, and then it goes on over here. So it is an insertion. And what you hopefully can appreciate is that the insertion is essentially the opposite or the substrate of the excision. Let's have a look at the last example over here. In this case, we have the two recombination sites facing one another. So let's start again by drawing the synapse. We have recombination site 1 and recombination site 2. We can draw the direction again, and now we need to connect the strands, and it will go like this. And we can also indicate the directionality here of the sequence. So it goes like this. And now we reconnect the strands. And if you follow it now, what you will realize is that this is an inversion. And we already talked about that from the excision an insertion follows, and from the insertion an excision if we repeat those reactions. But from an inversion, what follows is again an inversion. So the second reaction will be an inversion, the third will be an inversion, and so on. Let's have a look at two special cases that might be interesting. The first one is we have the same reaction as here, but we look at the plasmid and we have a recombination site facing in the other direction with this genomic locus again. What the result will be is essentially the same as here, but the sequence that is inserted will be looking in the other direction. And the second special case is, what if you invert both of these recombination sites? So if it looks essentially like this, the result should be clear. If we invert both sides, the directionality is the same again, because we said directionality is actually just a convention, and it only matters what the direction of one is in respect to the direction of the other. And so also this assembly here will be an inversion, just like the upper reaction. So now that you are an expert in predicting which outcome we'll have in all of these cases, we need to think about another aspect. We have seen that each of these reactions also has the reverse reaction. And after we excise the particular product, it could also insert again. And once it's inserted, it could be excised again. So it's a cycle, it's a chemical reaction. And so we need to ask ourselves which of these two states is more likely. Because sometimes our aim is to remove a sequence, so excise, and in other cases we actually want to insert something. So we need to think about which one is more likely and are we actually going to get the product that we desire. And we can think about it in two ways, which is thermodynamically and kinetically. So we treat this here as a chemical reaction, and in terms of excision, the end product will have two molecules instead of one. And it's the opposite for the insertion. So thinking about thermodynamics and entropy, the excision is more likely or more favorable. And if we think about kinetics, we have to think about what is more likely. So when we think about the insertion, there is essentially a plasmid in the nucleus that is floating around and is looking for a single sequence, a 34 base pair sequence, usually in billions of other base pairs, as for mouse or human, which is a very daunting task. But if we have the excision, what essentially needs to find one another is just two sequences that are maybe separated by 1,000 base pairs, or depending what the sequence in between is. So clearly, the excision is not only more likely thermodynamically, but also kinetically. So in other words, what we need to do is we need to make this arrow smaller to indicate that the insertion is very unlikely. But there is a trick that we can use to favor the insertion over the excision. So what we can do in order to favor one outcome over the other is to mutate the binding sites, to destabilize them. And how this works is we can, for example, take one of these recombination sites and we mutate them on one of these binding sites. So one binding site is mutated. And we can do the same for the other recombination site right here. And we call these special recombination sites, in this case, LOX71 and LOX66. And we can also now follow what will happen if we again draw the insertion reaction. So we have mutations here and here. I hope we can see this. So just as up here, and once they recombine, what we will get is a fully mutated recombination site and a wild-type site. So this is the LOX72 variant, and this is simply LoxP. And so while the single binding site mutants can still undergo recombination, the double mutants, so the fully mutant recombination sites, cannot. And in this way, usually we would have the reverse reaction again, but this cannot happen because the LOX72 is essentially inert. We could use the same principle also for the excision reaction. So we could mutate the left side over here and the right side over here, and we would get again a fully non-functional mutant and a wild-type LOXP site. However, this case is usually not done because we learned that the excision reaction is so much more favorable than the insertion reaction, so that we actually don't need the mutants here. But for the insertion reaction, this is highly relevant because we would never get the insertion product without the LOXP mutants. Today we learned how to draw a synapse that is formed by Cre or flip, and how this anti-parallel alignment allows us then to draw and align the different strands and reconnect them so we get the final recombination product. We also learned that we can use mutants of the binding sites to favor one reaction over another. Thanks for watching, my name is Felix, and see you in the next video.